Alright, hi guys, this is your anatomy exam podcast, and I've never done this before, so this should be a lot of fun. You guys should have the slides, they're on Blackboard, and what I think I'll do is I'll do chapters 1 through 3, stop it, chapters 4 and 5, stop that, and then do chapters 6, 7, 8, and 9, and then upload those 3 videos. So the exam is going to be 103 questions. About 80 of those are multiple choice, and then the rest are going to be fill in the blank. And you guys will have a word list to pick from for that, so that should be helpful. But keep in mind that learning structures, there's a lot of questions that are based around your knowledge of different parts of the body, or specifically bones or cell types or things like that. So it is, it's a pretty comprehensive exam from what we've done. We've covered a lot of stuff in the first 9 chapters. I realize that. So it's hard to get detailed on any one thing since I'm asking you to know so much material. Just keep that in mind while you're studying. So, starting with chapter 1, it's not a lot of detail here. Just be familiar with how we use terms and how words are broken down and how we name things, how we used to name things, and how we name things now. And of course, you should be pretty familiar with what anatomical position is. And just as a review, keep in mind the planes and then also the orientation of the hands, which dictates which side is dorsal and which side is, um, well, dorsal and ventral is the same thing. Which side is dorsal and which side is not dorsal, and the word is escaping me. So there you go. The different sections would be good to know, and then I really should have said anterior and posterior back there. So knowing these terms, they're woven into the questions. So you'll read a question, and and instead of saying something is in front of the body, I'll ask, is it anterior to a certain structure? So this should just be something that you guys have come to know now. So we've talked several times about how we talk about the brachial region instead of the arm. The antebrachium is the forearm. The only exception really is we never say the crural region for the leg. We always just say the leg. And I know that kind of stinks, but that's kind of how the world works. So there you go. You guys, it would be good to be familiar with these different regions: right upper quadrant, left upper quadrant, epigastric, umbilical, hypogastric, etc. You likely will have a question that asks you to identify something in one of these regions. And then kind of ending the chapter, it's going to be things like you're going to want to know parietal versus visceral, and then what is in between those, and finally, related to those, understand what potential spaces are and where they may exist that we've talked about in the body. That's a really quick run through of chapter 1. But again, it's things that keep popping up over and over again as far as terminology and etc. So chapter 2, it's where we started talking about cells, and you guys are pretty familiar with the different types of epithelium, and you're going to need to be for the exam. So just be able to identify the difference between, simple squamous, and of course, know some examples of each, specifically lungs, arteries, and veins for simple squamous. We've talked about stratified squamous, and we'll talk about it in more detail in chapter 3. Remember that stratum lucidum is optional, if you will, or it's only exists in thick skin, not thin skin. And I would be able to identify what stratified squamous looks like, and also know that the stratified squamous exists in the esophagus and the vagina, and then know the difference between keratinized and non-keratinized. But I think we'll cover that again in chapter 3 here in a second. Cuboidal, pretty self-explanatory. Simple columnar, again, you can see the entire cell, and contradict that with pseudostratified columnar, whereas it looks like there's multiple cells stacked on top of each other, but really what you're seeing is different sections of the cell. And also, I would know some examples of where you find these. Just a very straightforward summary of the differences of the types of cells, and again, we'll see that in chapter 3. And then I would be familiar with different types of transitional sections. So this is the esophagus, where we go from stratified squamous to columnar, and I wouldn't worry about any specific pathologies, but I would just know that areas where this can occur that we've talked about in class, as well as maybe some side effects or some possible complications, I should say. Now, when we talk about a cell, I think we've used these terms enough at this point that you should be pretty familiar or comfortable with the apical side, basal side, basement membrane. Hemidesmosomes, they connect cells to the basement membrane. Desmosomes connect cells to each other. And then microvilli, they do things like increase surface area for absorption. Make sure you don't confuse those with cilia or flagella. I think we will see a slide later on that kind of differentiates those three things. And then we have different types of cell junctions. There's tight junctions, which seal the area around the cell so fluids can't pass through here. There's desmosomes, which are really the force that holds cells together. And then there's gap junctions. This is how cells can communicate. And one of the more prominent examples in anatomy would be gap junctions between cardiac cells and the heart. That's how the contractions are synchronized. We talked about two diseases related to dysfunctions, if you will, in cell connectivity. These are both autoimmune diseases, pemphigus vulgaris and bullous pemphigoid. You don't need to understand that pemphigus vulgaris is autoantibodies against desmosomes, and you don't need to understand that bullous pemphigoid is autoantibodies against hemidesmosomes. You simply just need to know that they are blistering diseases that involve the epidermis of the skin, or I should just say the epidermis, really. Membrane proteins. The key here is that you understand there's different ways for materials to enter and leave a cell. So simple diffusion is, I'm going to say the simplest and probably regret it someday, is the most straightforward one. And it's simply when materials either diffuse directly across the cell membrane or through a channel, and it doesn't require any energy. And facilitated diffusion, this molecule this molecule doesn't necessarily just float through. It actually has to come into a receptor and then be released in the cell. You'll notice that none of these are actually diffusing just through the membrane. And then finally, active transport. The important idea here is that this requires energy. So the other two types don't require energy, an energy input from the cell, but active transport does. And usually it's because you're moving from an area of high concentration to low concentration, or low to high, I should say. This is excreting something where there's a high concentration in the cell, whereas if this was, if this molecule was allowed to move freely, it would move to areas of low concentration. Okay, so here's the microvilli versus cilia versus flagella slide. Remember that flagella, really the only place in humans we find it is in sperm. Cilia, these are, you can imagine cilia, I'm congested right now, and what's happening is the cilia in my lungs are moving mucus up and out of my lungs. So, but microvilli, they're not tools for moving things. They're tools for increasing surface area absorption. So those are the key differences between these three. We went through a lot of organelles. Some ones that I would focus on if I were you. We looked at endopath, the ER, and I asked you several times throughout the past few weeks examples of what is made in the ER. So focusing on the red here would be a good idea. Again, here's just an image of the ER. Ribosomes, they are attached to the ER, and they're also free in the cytoplasm. And where they are dictates where the proteins they make end up. And then the Golgi complex, just remember that it's a transport system. So the ER is going to, just slide right there. The ER will send proteins to the Golgi for further processing and then trafficking. So they may stay within the cell or they may be excreted outside the cell. Lysosomes are simply a way to consume either materials brought in from the outside or materials brought in from the inside. They're also involved in apoptosis, but I wouldn't worry about that for this exam. And then finally mitochondria, which they're the energy producer of the cell. So if you don't have mitochondria, if you have mitochondrial dysfunction, certain activities that we talked about a few slides ago, you might have problems with that. So keep that in mind. The cell cycle, it would be good to know just an overview of what happens during G1, what happens during S, what happens during G2, and then you should be pretty familiar with what happens during mitosis. I will not ask you specifically the order of mitoses. If you tell my advisor, I'm sure he will disown me forever, but I'm not going to worry about that. I'm more concerned that you know G1S, G2. And here's just a summary of that. And here's me telling you I won't ask and making a crappy joke. And finally, stem cells. I think you guys are comfortable with this at this point. They replace they replace essentially nearly every tissue in our body. So chapter 3 is the histology chapter. So we go into a little bit more detail about different types of epithelium, again, just to be able to identify these based on the image, and also a general idea of why you would have simple squamous in one place and why you would have cuboidal in one place, for example. So again, these are just for really straightforward identification. We talked about keratinized versus non-keratinized stratified squamous. In chapter 5 in the integument, I'll go into more detail about the connective tissue in here. Just for now, if you're in chapter 3, make sure that you understand the difference between the two and also examples of where you would find keratinized versus non-keratinized. Stratified cuboidal, very straightforward. Transitional, we didn't really talk about this a lot. We use transitional in a different way in a pathologic sense, but just not to confuse you, there are transitional epitheliums. Some are in the bladder, some are in the kidneys, or classically, I should say, that's the best examples people use. But again, what we talked about was transitional in a pathologic sense, where the cells transition from one type to another. Okay, so connective tissue. This can get confusing, but it will be worth it for you guys to know the difference between different kinds of connective tissue. Remember that there's 3 types of fibers: collagenous, reticular, and elastic. I would know some examples. I would know examples of each, and I would also understand the difference between these 3. If you look at loose connective tissue, there are 2 types: areolar and reticular. Reticular specifically, think of it in terms of lymphoid organs. Okay, this is a spleen, and it's friable. It's not very. You can tell by the amount of fibers in here that it's probably not very strong. So it's true for your lymphatic organs. Areolar, the key here, you're going to see it in the papillary layer of the epidermis. So understand that difference. We have 2 types of dense connective tissue. We have dense regular and dense irregular. I think just from the image, it's obvious what the difference is. Again, I would know where you find these. So, a ligament or a tendon, dense regular. Dense irregular, you're going to find in certain layers of the dermis. Adipose tissue, just another type of tissue to be familiar with. Then we have cartilage. I wouldn't worry so much about being able to identify based on images cartilage. I would simply understand some examples of different types of cartilage: elastic fiber cartilage and hyaline, where you could find those. When we talked about bone, you guys remember that we talked about chondrocytes. Osteocytes. Here we talk about chondrocytes. Those are the mature cells in cartilage. So bone we'll talk about a lot more in chapter 6, but I would be familiar with an image like this. Blood, not really going to come up on this exam. We'll get into that later in the year or in the semester. Now, muscular tissue, I would understand the difference between these 3. I would also be able to identify these 3 based on the image. The key identifying or differentiating them visually is if you look at skeletal muscle, it has striations. It looks a little out of focus. cardiac, the striations are the connections between the cells, and the cells are connected to each other. And there's something in between these cells that is important for their communication. I would understand that. And then again, smooth muscle, um, it's not striated is typically how I would identify it, and it doesn't look like cardiac tissue. So, and these are intercalated discs. That's what's between the two cells. So this is a good slide to spend a little time on. Glands comes up here and there. We'll talk about different types of excretion, or secretion, I should say. Actually, I think next. Yeah, so meiocrine glands, they they excrete through exocytosis, and here's some examples of that. Apocrine glands, I wouldn't really worry about this. It's not really used as much anymore. And then holocrine glands, understand how they do their secretion. And here's just an image of that. As far as tissue growth goes, starting with the changes, metaplasia, it is reversible. So we talked about transitional epithelium. That's a form of metaplasia. And then you have different types of growth. Neoplasia is pathological, but cells can just multiply and not be cancerous. But cells can also just get bigger. So hypertrophy versus hyperplasia. Okay, I'm going to stop it here, and then I'm going to pick up again with Chapter 4 and Chapter 5.